Hello viewers, welcome to my channel of biotechnology tutorial and research lab. I am Associate Professor Dr. Sharif Hussain, obtained my PhD in biotechnology from Ehime University, Japan. The title of my presentation today is very interesting. This is bioethanol production from fruit biomass as antiseptic or sanitizer and anti-fermenter. You can see here we used different types of fruits, rotten date fruits. This is the rotten grapes, and this is the rotten banana fruits. So before starting my lecture or presentation, you are requested to subscribe my channel to get updated and innovative data in future. Let's move. Details title: Bioethanol production from banana, grapes and dates fruits. Biomasses as antibacterial, means antiseptic or sanitizer, and biosolvent, means anti-fermenter. Its physical, chemical and biochemical properties. Significance of the presentation of the research. You can see here bioenergy can be defined as energy obtained from biomass, which is the biodegradable fraction of products, waste and residue from agricultural, like botanical, vegetables and animals origins, forestry and related industries. Also from the biodegradable fractions of industrial and municipal waste. Different forms of bioenergy can be produced from a wide range of biomass sources, for example, agricultural residues like fruit waste, vegetable waste, crops waste, and different types of the wood waste. This is the II, infrared radiation carries greenhouse gases such as water vapor, carbon dioxide, ozone, and the methane, delay its eventual escape into space. The atmosphere is warmed by this mechanism and in turn emits IR radiation with portions of this energy could warm the surface and lower atmosphere. As a result, the temperature of the earth would maintain about 33 degrees centigrade higher than it should be without atmospheric and the radiation of IR energy. So, however, problems may arise when the atmosphere concentration of GSG increase, means greenhouse gas emission increase. One of the causes of this problem is because the burning of the fossil fuels. When it is burnt, it could increase the rate of carbon dioxide, carbon monoxide, and sulfur and carbon monoxide. Energy consumption around the world has increased sevenfold in the last century. As a effect of the burning of fossil fuels, mainly used in transportation sector, caused the primary atmospheric pollutions, oxidations of carbon dioxide, sulfur dioxide, and the nitrous oxide emissions. Combustion of the fossil fuels at the current rates would contribute to the environmental crisis globally. The increase in demanding fuels combined with depletion of reserve minerals oil has led to the development of eco-friendly concept. In addition, demand of the energy increases with the increase of the world population and urbanizations. And thus, development of bioenergy, biomaterials, and bioproducts as alternative energy or alternative resources might keep to reduce these problems. Different fruits waste have been used to produce biogas using fail-based digestion in Australia. In order to develop the new technologies, improving the available technologies regarding the biofuel productions. In order to develop technologies and improving the available technologies regarding biofuel productions, it is essential to address the challenge and opportunity of the biofuels in the context of food security and sustainable development needs. It was reported that ethanol production by fermentation faces competition with ethanol production from petroleum-based waste product. However, as the value of the petrochemicals were increased, fermentation of ethanol received more attention since renewable materials waste products are cheaper, sometimes nothing to pay. That is why it is easily available and more economical. So biofuels or biomaterials give more benefits since it comes from renewable energy, sustainability, reduced greenhouse gas emissions, regional developments, social structure and agricultural and security supply. Pineapple waste had potential for recycling in order to get valuable raw materials. Convert into useful and higher value products, foods or food after biological treatment, even as even as raw material for this industry, for other industries. One of the example of the raw materials is pineapple waste that is converted to the bioethanol productions. The objectives of this study were to investigate the influence of the different temperature shaking hours, yeast and enzymes concentrations on bioethanol productions by using rotten banana and, and dates, and then the grapes, rotten banana, grapes and dates, its pulp, peel and also its victor seeds. In addition to know the standard properties, viscosity, pH and the metal content of bioethanol for the use in the petrol engine. Also, the other objectives is to produce the sanitizer or antiseptic from this bioethanol uh, as antibacterial treatment, and secondly also this is called anti-fermenter to protect the foods or foods materials from the rotting. So this is good topic. uh, so we can see now materials and method, how it has been done or it has been produced. Experiment 1, banana waste. The banana waste were brought from the market, and then this experiment my MSc student did in Malaysia. In Malaysia, the enzymes used in this experiment, different types of enzymes, amylase and cellulase enzymes, and then Aspergillus niger, cellulose name, yeast derived from the culture of the Saccharomyces yeast are used. So, and also different types of the buffer solutions and diet and also others conditions. This is a we have collected it from the Sigma companies. Preparation of samples. 100 grams of rotten bananas were thoroughly washed with distilled water, cut using sterile knives and are blended by using the sterile automatic juice blender. The banana mass was then dispensed into the total of 9 sterile spot bottles, where 3 replicates for each sample. Then we use 25 ml of water were added into the spot bottle containing banana mass. pH content of the was maintained, and after that total soluble sewage of banana mass were taken. Then fermentation technology, the methods 1, 3 and 5 gram per liter of yeast, Saccharomyces cerevisiae were added into each set, and all of the bottles are closed to ensure they were made airtight to provide and anaerobic conditions and placed in incubator at 28, 30 and 35 degrees centigrade. The dry agri yeast were hydrated in the water bath at 40 degrees centigrade by using clean water allowed taking to room temperature before added into the banana mash. Fermentation was carried out for 5 days, 0, 2, 3 and 5 days. After fermentation, the clean distill cotton cloths were used to sieve the product from the residue. Extract were collected in 9 different sterile plastic containers, and then the obtained raw bioethanol was then taken in room temperature to measure pH and total soluble solids. The bioethanol yield was measured from the fermentation of rotten bananas mashed by using different shaking hours 0, 3 and 6 hours. Sample obtained from different fermentation processes were determined by the change of pH, total soluble solids and bioethanol concentrations, and residue weight, viscosity and elements analysis was done. Change of the pH of all the fermentations were determined. The pH was checked before and after fermentation process. Total soluble solids content of all fermentation was determined by using ATAGO digital refractometer. Ethanol concentration was determined by ethyl alcohol refractometer. Japan glucose content was determined by glucose digital refractometer. Japan. Experiment 2 grapes. The same procedure the procedures was followed, but we can just sample is difference. Preparation of the sample was same as experiment 1 except raw materials. Here sample was used as rotten grapes. Fermentation was done by using the following parameters 0, 24, 74, 72 and 120 hours, as given as 135 gram per liter is concentration. Ethanol concentration was determined by ethyl alcohol refractometer. Same as experiment 1, glucose content determined by glucose digital refractometer, same as experiment 1. You can see here, this is the dead samples, rotten dead samples, which is not useless. It is not waste dates, not eatable, not edible. So this is the also rotten, and this is the paste, dates paste, and then after dates paste, this is the then this is incubator in the Scott bottle fermentation techniques. In the fermentation, you can see here. After fermentation, this is filtration was done. After filtration, the taste different multi-oil analyzer, and also this is the others for TSS meter, and then this is the raw bioethanol. This is the bioethanol, and then this is the refractometer. So this is all about the process, filtration process, and also incubation fermentation process. Bioethanol for total soluble solids contain bioethanols and the ethyl alcohol device. This is process. The experiment 3 also dates, fruits, waste. This is the we can see here the experiment 3 date, fruits, waste for experiment 3 1. So preparation of samples are same as experiment 1, except raw materials. Here samples were used as rotten dates, fruits, waste. Rotten dates, you can see here rotten dates, fruits, waste. Here this is rotten dates, and rotten dates we used in the experiment. And then here we can see done and followed the 0, 24, 72 and 120, and then hours as well as 135 gram per liter is concentrations, and the others are same. Ethanol concentration was determined. Ethanol refractometer as same as experiment 1. Glucose content was determined. Glucose digital refractometer same as experiment 1. Disinfectant experiment as antiseptics or sanitizer. Nowadays it is most important. We have produced sanitizer from waste fruit materials. This is most important for antibacterial and antiviral treatment. Disinfectant experiment. The experiment was performed in 1.5 tubes, 3 different contact times, 5 minutes, 10 minutes and 15 minutes were also tested for these tubes. You can see 0.1 ml of cultural solution was added into 0.9 ml of disinfected After certain contact time, we can see uh, uh, 5000 rpm centrifuge was performed for 5 minutes to separate the culture from the solutions. So this supernatant was discarded, and then the tube was refilled by deionized water, followed by separate plating on each tube. This is bacterial culture. Bacterial culture we have done. My institution done to see the disinfected experiment. After the experiment, all the results tubes were stored in the refrigerator at 4 degrees centigrade, and the next day plates containing was performed on each spread plate after 24 hours culturing at 37 degrees centigrade in the incubator. Use of bioethanol and the biosolvent and fermenter. Grape juice was used to test the produced bioethanol as biosolvent, whether it is anti-fermenter or not. Juice was kept for 4 days, mixing with bioethanol and without bioethanol. Control. 5 drops of bioethanol were added with the grapes juice and observed its rotten condition at room temperature. Glucose content and bioethanol percent were measured from 1 to 4 days. What is our discussion? We can see here. So almost a concentration of the bioethanol different shaking hours was shown in figure 1. The fermented banana mass that had been shaken for 6 hours produced higher bioethanol with 6.55 VB volume by volume, followed by 3 hours of shaking period and fermentation of banana mass without shaking. This is the most result. The concentration of bioethanol increased and the time of shaking process was increased. So also after that, the data, the values of the total soluble solid for the fermented banana mass were lower than before fermentation. So here also glucose analysis table 2 showed the glucose prepared by using digital spectrometer. There was a rapid decrease of glucose concentration from 0 to 24 hours one day of fermentation period, and the glucose is utilized by yeast cell to produce bioethanol. However, from 72 hours to 120 hours days, there was only slight increment of the bioethanol concentrations from 5.86 to 6.1. The large amount of glucose was utilized at the initial stage post the rapid bioethanol productions within 24 hours, where 5.51% of bioethanol was produced. The highest bioethanol produced and the lowest glucose concentration was observed at 120 hours, where concentration of bioethanol was 6.1%, while glucose concentration was 0.52%. Experiment 2 almost same also. Glucose content was determined by digital alcohol spectrometer. The sugar using sugar concentration have been evaluated from the samples of fermentations of the grapes. Then you can see the different types of pH and 30 degrees centigrade, pH 5, and different hours of incubations. Then we can see from the figure 2 the sugar content measures are decreased at the fermentation going on, and the ethanol production increased. The viscosity of the bioethanol produced was important when considering the spray characteristics of the fuel within the engines. Others we can see here. This is also all the data was AS10 standard we have done. Then after that we can see different types of the results in experiment 3. Here also the effect of initial pH maximum yield of bioethanol was got. The 5.5 to 6 oscillated and other fermentations fermenter were kept constant level. So here you can see here the yeast component suffers from various stages during ethanol fermentation, such as nutrient deficiency. So here you can see different types of bioethanol solvent and antiseptic. This is the important. From Feb 12, it has been seen that glucose content was started to reduce the first. After 12 hours, and bioethanol was started to produce and made rotten the juice faster in the grapes juice without produced bioethanol from dates at room temperature. Juice mixing with bioethanol showed glucose content was stable for 2 days. When we mixed the bioethanol, 2 days it was stable, and then from 3 days it was started to rot slowly, and bioethanol production was lower than control. It might be due to the bioethanol effect on the juice as solvent. That is why rotten started in the juice after 2 days earlier. Then, then glucose content reducing as well as grapes bioethanol productions in rotten conditions made faster in the control without bioethanol. Then in the juice with dates bioethanol. From this result, it can be suggested that bioethanol also can be used as antiseptic, means uh, effect of sanitization and laboratory use, and antibacterial or antiviral use. You can see here the result just before we discuss about banana, grapes, and the dates. So this bioethanol yield, you have found the highest bioethanol in the dates. The dates 18 compared to grapes and banana. Also others parameters also. It is under and glucose content also. They found the good before and after. In case of dates, here also we can see the samples parameters, different temperature, dates, grapes and banana. The highest one we have found in the dates compared to the banana and grapes. Also others also. The highest was found in case of the dates compared to the banana and grapes. Here we can see the correlation of glucose and ethanol percentage from the dates by biomass treated with different fermentation period. When the bioethanol was decreasing, you can see glucose production was decreasing, bioethanol production was increasing. That it is bioethanol increasing and glucose decreasing. This is the correlation. The correlation between glucose and ethanol. You can see here the fermenter. First 1 day, first day, second day, third day, fourth day, glucose concentration controlled by ethanol. Which is first day, and here the glucose concentration is it means the glucose first, second, third, fourth. It means bioethanol productions. Bioethanol productions is increasing and glucose production is decreasing. Glucose production decreasing. So this is the control also here increasing. Control is higher than bioethanol. So here also you can see disinfection. You can see here in this parameter, in this experiment. This is called the experiment of the different types of the antiseptic experiment. Measure here. Okay, this is how much bacterial colony or culture by bioethanol as antiseptic or as sanitizer. Sanitizer, how many bacteria has been killed. So you can see here different types of different here you can see the disinfected bioethanol. Yeah, this is the topic. This is the topics and the amount of this is the export times, disinfected bioethanol banana, bioethanol grapes, bioethanol dates, and then control. This is control. You can see here. You can control it was the highest date, but in the bioethanol banana, bioethanol grapes and dates also there is the banana is the 5 minutes, 10 minutes, 15 minutes, 20 minutes. You can see here the 15 minutes. In the grapes a little bit less bacteria has been killed, and in dates also less, but the highest is banana bioethanol. killed a lot of bacteria. Yeah, so this is the it has been proved that it can be used as the antiseptic sanitizer from the our result, laboratory result. Determination of the viscosity and acid value determined to see the how much is viscous, and then acid content, and then determined of the chemical content, different types, copper, lead, ferrous, silicon, phosphorus, copper. It is all ASTM standard, all ASTM standard. So this is all about the result. Conclusion: it can be concluded that production of bioethanol from fruits biomass is an interesting issue that can be used as environmental recycling process for waste management. First things. Then second, sanitizer or antiseptic made from fruit waste can be used for antibacterial and antiviral treatments. Anti-fermenter made from fruit waste can be used to protect the products from rotting. So this is best important two new things: sanitizer and the anti-fermenter. Bioethanol can be used. In addition, production of bioethanol can be used for the fuel purpose also. The list of applications, the references of the papers. Our names also there. We published many papers regarding this topic. Okay, so thank you very much for listening, watching, and subscribing our channel. Thank you again.